I can hear.
Okay.
Yes, Abhijit, go ahead.
Okay. So good morning, everyone. This is in continuation with our scientific talk with respect to this Science Setu program. Today, I'm very pleased to introduce today's speaker. Today, Dr. Hegde, which is scientist H at NIB, is going to deliver a talk on animals in human welfare, not just milk, meat, but also maladies. So we are also celebrating this 75 years of post-independence, that is Azadi Ka Amrit Mahotsav. So this talk is also under Azadi Ka Amrit Mahotsav uh, with respect to the Sashakt Bharat and Vigyan Ke Vikas Pradyogiki Se Pragati 100. So let me give a brief introduction of Dr. Hegde. Dr. Hegde is an animal virologist, immunologist with experience in veterinary and medical virology, immunology, cell biology, and microbiology. He did BVSc and MUSc, BVSc in 1989 and MUSc in 1992 from University of Agricultural Sciences, Bangalore, India. After that, he moved to US for his postdoctoral studies. He did his PhD in 1998 from University of Nebraska, USA. After that, he was there for several years as a postdoctoral fellow and uh, research associate from 1998 to 2004 at Oregon Health and Sciences University, USA. So there he studied animal virology, health, post-pathogen interactions with respect to the viral diseases. So after that, he came back to India and joined Ella Foundation in 2006, initially as a group leader, and then he became associate director from 2014 to 2017 at Ella Foundation, Hyderabad. Dr. Hegde joined NIAB in 2017 as a scientist G and became scientist H in 2020. So at NIB, he's working on different aspects. To name a few, he's working on mastitis, antimicrobial resistance, zoonosis, One Health, ephemeral viral virus. He has, with respect to the awards, he has received several national international awards. He is academic editor of Class 1 Clinical Vaccine Immunology, now MSPEAR. He has also contributed to the development of World Health Organization draft guidelines on quality, safety, efficacy of Ebola vaccines 2015. He has published more than 60-70 papers, more than 15 reviews. He has one international patent, US patent on chikungunya virus. He has filed several patents that he has more than 2,000 citations. He received more than 2,000 citations for his publications. So now I invite Dr. Hegde to deliver his talk on animals in human welfare, not just milk, meat, but also maladies. I hope you will enjoy this talk. Thank you.
Thank you, Abhijit. Can you hear me?
Yes, sir.
Okay. So it's loud enough, or should I increase the volume?
It is loud enough, sir.
Okay, fine. So thank you for that kind introduction. So today's topic is a little bit between what animals contribute to human health and welfare, and also in terms of how our health can be disturbed through animals. So I'll start sharing my screen. Can you see? Is it full screen?
Yes.
Yes, it's fine.
Okay, so the title, as has already been introduced, is Animals in Human Welfare, not just milk and meat, but also maladies. I know this is mostly for college students, so if you are aware of some English, you know what alliteration is. That is words starting with the same letter, and in this case, milk, meat, and maladies. You may already be knowing, or you must know, that humans do derive food in terms of milk and meat from animals, and of course, other materials, which I'll come to in a little bit. But they can also transmit certain diseases, and maladies is nothing but ill health or diseases. I have put up two books which have the word maladies in them. One is on the left side, if you see, The Emperor of All Maladies. This was written by Siddhartha Mukherjee. He is a doctor in US, and he wrote about the biography of cancer. So if anyone is interested in what is cancer and how the treatment of cancer evolved from the olden days, this is one of the books that you should read. The other one is the 1 minute. So the other one is The other one is The Interpreter of Maladies by Jhampa Lahiri. This has nothing to do with diseases, actually. This is a collection of short stories which won her the Pulitzer Prize. So with this introduction, I'll go a little bit into the topics that I want to talk about. So the first slide I put up here is just to remind me that this is part of the Science Setu program. So on top, you see all these things that remind us of science, be it physics, chemistry, or biology. And then on either side, I have two bridges, the Setus. This is actually a bridge in China, which reminds me of the shape of the DNA, if you will. And on the other side, here is another bridge. This is actually not a two-looped bridge. So here you see the line of water, and this is actually the reflection. And why I have put this slide is this looks like the DNA. And if you remember, from the DNA, you produce RNA. Or you could even imagine that the DNA is being replicated. So this is the mirror image of the DNA. And I'm fond of one of our own Indian cartoonists, Mr. R.K. Lakshman, who once imagined that we need to even have traffic lights in space. So just to introduce you to the program that Science Setu, where we do want to reach out to other people and students to engage in science. So I'll come to the specific topic that I'm going to talk about today. One, initially, in order to understand animal science or the productivity of animals, we need to understand how far have we come in agriculture and related sciences. So man began to, was initially a hunter-gatherer eating only raw food. And only when man could fire or start a fire was cooking started. But since then, the real start of agriculture took a lot of number of years. So basically 1.9 million years ago, the fire was invented, but then it took almost that much same time in order for us to start serious agriculture. If you see here, the development of agriculture, use of grains in the diet started only 9,000 years ago, but the use of the modern flour was only 200 years ago. And the serious agriculture started barely in the last 50 years in this timeline. So what happened in this duration or timeline in the in terms of agricultural evolution? We started with hunter-gatherers, as I said, and then there was cooking. And then in the second agricultural revolution, we started with making nalas or irrigation, and also using certain improved flows, including certain mechanical ones after the Industrial Revolution. And then there was a huge revolution called the Green Revolution, which is actually the third agricultural revolution. Originally started by this person called Norman Borlaug. He was a US scientist, and within India by M.S. Swaminathan. So what started after that is the use of hybrid varieties, use of fertilizer, and then nowadays we see this plants or crops being produced in intensive way in polyhouses. The fourth agricultural revolution, which we are witnessing right now, will be the use of Internet of Things or connecting the farm to control everything. So you have drones, you have sensors, you can measure the variables such as the temperature, moisture, the pests, and things like that. And also you can have a polyhouse where you can control a lot of things. So basically you can control the agricultural production through electronic devices. So this is where the agricultural revolution is going right now. On the side, the associated aspects of agriculture, we also started farming animals or taming wild animals. All these pictures of animals that you see here were originally in the wild. So the earliest domesticated animals were, of course, our own good friends, the dogs, which is 15,000 years ago, which was followed by sheep, goats, cattle, pigs, and various other animals, including the latest ones, the birds. And all these have been used for food as well as other products that we use in our daily lives. There was also another revolution called the White Revolution. And this is the picture of Verghese Kurian, who brought in the White Revolution in India. And his simple motto was 8 hours of dairy, 8 hours of family, and 8 hours of sleep. So basically, he wanted to make sure that India was self-sufficient in milk production and would get enough protein through milk. So how did that come up actually? So if you look here in the 1950s, both the production, which is shown in these bars here, as well as availability per person was low. And it started slowly shooting up in the late 80s. And now we have a huge production of milk as well as availability of milk per person. So what did he really do? He basically started what are known as milk cooperatives. So in the Western countries, typically they have large dairy herds and a lot of mechanization where you have hundreds of thousands of cattle that can be milked in a particular area. And they don't really have this kind of a setup. In India, the problem was most of the milking animals were held in small holding where people would have, 1, 2, 3, up to 10 animals. So somehow these animals products such as milk needed to be collected and then distributed. So he started what is known as the cooperative operation. So this started from the villages where you would collect it into a local village. From there, it would go to the district milk union and it would go to the state milk federation. From there, the milk is distributed and the revenues are flowed back to the farmer. So this is the cooperative milk unions that are operating in India even today. And the milk you get is mostly through this cooperative union. We also went through what we call as a red or a pink revolution. This is basically meat production. As you can see, India leads in the export of beef, or it used to lead in the export of beef a few years ago, and in the world. And now we are actually coming up in poultry meat production. And these are not only good source of protein for the country, but also we export them. So in nutshell, India is #1 in number of buffaloes in India, #2 in number of cattle and goats in India, #3 in sheep and fish production in the world. And also we are #4 or 5 in poultry production. This is as pertaining to the number of animals or fish. But if you look at the productivity, India is #1 in the world in the total milk production. And it's actually #2 in fish production. And it is #3 or 4 in the number of eggs being produced. So we produce a lot of food products through animals and birds. But that's not all. Animals are also involved in various other ways to improve human health and welfare. Food is what I have already talked about. They provide milk, meat, and eggs. But we also obtain skin, which is used to produce hide, wool, wool, which is produced to used to produce coats and things like that. Hair is used, and we also use felt. So this is all useful in one way or another. They are also used for draught purposes, including bullocks, horses, ponies, and in desert places, camels. Manure is another byproduct where in villages you may have heard that biogas production is also there. And of course, especially sheep and goats can be used in weed control when they are sent out for grazing. There are examples in the world where certain weeds have been greatly controlled by grazing. They are also used for recreation, for example, horse racing, and even in India, you may have heard about this Jallikattu. So bull racing, that is also seen in India. And of course, cats and dogs, which are our companions. So a lot of people, especially in the cities, have cats and dogs and companions. So these are all the different ways that animals contribute to our health and welfare. Welfare, I mean, great example is dogs. Dogs are good companions that provide us pleasure, and also, they are there when we are sad. So in order to actually have good livestock productivity, it's not sufficient for us to do one thing. We have to actually have a 360-degree application for livestock productivity. That involves control of diseases, and I'll come to some of the zoonotic diseases in a little bit, and improving their nutrition, be it identifying new strains of grass, or fortifying the grass with certain nutritious metals or chemicals or other things. Human factors like religious beliefs and tradition. For example, they worship the cow. Market demand for milk, meat, or other products, availability of necessary production inputs, veterinary care, supplements, including feed supplements, etc. Climate and geography plays a role, because we can't have all sorts of animals in all sorts of climates. And in India, we have very varied climate, ranging from heavy rain to totally dry areas. And of course, parasites are a problem, because a lot of the Indian livestock are sent out for grazing. So all these actually have a complex interaction in livestock productivity. And besides all this is genetics and selection. I say this because earlier, mostly in the White Revolution, before the White Revolution, we were mostly using Indian breeds, which were not giving enough milk to be self-sufficient. But then later on, we introduced what is known as crossbreeding to improve the genetic makeup of our Indian breeds, so that they would yield higher milk. So this is one of the major things that led to our White Revolution. So now, having said all that, what is the input of animals in milk, meat, and other things in human beings? I would like to switch gears as to how they contribute to our maladies or diseases. These are just two cartoons that show that there are animals that can spread, there are bugs that can spread, bacteria, viruses that can spread to humans. So these things are the diseases that come from animals to humans are called zoonosis. So, and this is the major title that major part of the talk that I would like to focus on for the rest of the talk. So what is zoonosis? Zoonosis is derived from two words. One is zoon and nosis, but it's not coming from the combination of these two pictures, the zoo and the nosis. It's basically, what it means is transmission of diseases between animals and humans, not necessarily from animals to humans. It can also be from humans to animals. There are certain diseases that we pass on to animals as well. But ultimately, what we care about, maybe you can say that we are selfish humans, is that we care more about infectious agents that are coming from animals and then jump to humans and then are passed on between humans to humans. Any discussion with zoonosis or diseases that transmit from animals start from rabies. You may all be aware that, of or heard about mad dogs running around in the streets and biting other dogs, animals, or human beings. But that is just one of the zoonotic diseases. There are several other zoonotic diseases. And if you really see, there are more than 1,700 human pathogens identified. These are bacteria, viruses, parasites, fungi, all sorts of things. And close to 50% of them come from animals. There are 156 identified emerging pathogens, out of which almost 2/3 come from animals. And if you look at the emerging pathogens, which are 3 times more likely to be coming from animals than from humans alone. And there was, in fact, a study which showed that when they studied diseases from 1940 to 2004, a 64-year span, and they studied emerging infectious diseases, they identified 335 EIDs, out of which 60% came from animals, and of these, 70% came from wildlife, not even domesticated animals. So this is the graph that shows, if you see here, the white portion of these graphs is the one that is coming from wildlife. So a major portion of zoonotic diseases come from wildlife, not just domestic animals. Indeed, it's been shown that there is one new infectious disease identified every year for each of the last 30 to 35 years. And it's not concentrated to just the third world countries. If you look at Africa or parts of Asia, a lot of them are, in fact, identified in Western countries, where there is more intensive surveillance program or identification program. And if you look at the animal kingdom, there are more than 1.5 million species described, and 7 million of them are estimated totally, and 1 million of them are just insects. If you look at the other animals in this kingdom, so this is on the y-axis is into thousands. This is the number of species. So there are certain of them which are low in number, but there are a huge number of mollusks and a huge number of chordates. This is where most of the vertebrates are. So each of these could be involved in some way or another to give disease to human beings, whether it is they are intermediate hosts or they are direct hosts. Each of these could be potentially involved. And the additional thing is, in the recent times, what we call genome sequencing has really sped up the study of infectious disease. So what is genome sequencing? So you've all seen the structure of a cell, and it has a nucleus. Inside the nucleus is its DNA. And you probably all know that the DNA is wound around in these chromosomes. And if you disentangle the chromosomes, you see these beaded-like structures, which are nucleosomes. And then there are further these building blocks, so-called nucleotides. And the sequence of these nucleotides determines the species, determines the protein, determines the variability between different things. So this can actually be elucidated or identified. The sequence of this particular gene or the whole genome can be done, and you can go and figure out what species it belongs to, or even identify infectious agents. So we have, in the recent times, we have been able to do a whole genome sequencing of parasites, protozoa, fungi, prokaryotes like bacteria, and of course viruses. And you all know about SARS coronavirus. So there's a lot of sequencing of viruses that is going on. This genome sequencing has really sped up the study of infectious diseases. In what terms? I'll explain a little briefly in the next few slides. So genomics has allowed us to pinpoint the first case of a disease. So we can actually track back and identify who was the first person who spread this disease to other people. We can identify potential origin. So this may be coming from a wild animal or a domestic animal, so we can identify that. We can track the route of spread. So by actually looking at certain nucleotide changes, we can track from where to where the virus or the bacterium spread. We can even predict the transmission based on which way it is moving. We can study the evolution of the pathogen. You may have heard of the variants of concern or mutations in SARS coronavirus. And also we may be able to think about stopping because we understand all these. So one example here, the example of coronavirus or COVID. So this one particular mutation from the UK had spread to various parts of the country, and people have been able to actually track where it originated and how it spread. And similarly is the South African variant and the Brazilian variant. And now we are talking about the Indian double mutant. We can actually identify where the virus or the bacterium came from, what was the reservoir host. Index case is nothing but the first case. And we can actually figure out in some cases how it spread. Did it spread through an infected individual in an aeroplane, or did it spread through a vector or other means? Indeed, I give you some examples. This is the example of bird flu, the H5N1 influenza virus. So initially, the first ever outbreak of bird flu started in China, and it spread to different parts of the world. So basically in Guangzhou, then it went to Southeast Asia, Korea, Japan, as well as the Middle East. So exactly people have been able to locate how this thing spread. Of course, there's also additional example of pandemic influenza. This happened in 2009, originated first from Mexico, then went to the US and spread from US to other parts of the world. And one particular example I can give you where a passenger, a group of students and a teacher visited Ontario, and then they moved to Tokyo. From there, different students went to different places and they carried the virus to Beijing, Shanghai, Taiwan, and from there to Hong Kong. And basically, based on the genome sequencing, people have been able to identify how exactly this virus moved and how it transmitted to different parts of the world. Other examples, both of these are coronaviruses. This is actually SARS coronavirus 1, a close relative of the COVID virus. Started again in Hong Kong, and from there it spread to various parts of the world. And this is actually a Middle East respiratory coronavirus, so-called MERS virus, which started in Saudi Arabia and spread to different parts of the world. And of course, one of the deadlier viruses called Ebola virus, which up until the latest outbreak, the 2014 outbreak, it was concentrated mostly in the Congo and the Central Africa. But then this 2014-15 outbreak was in Western Africa. And basically, there were people who traveled to other parts of the globe, and there were individual cases or few cases, handful of cases in various parts of the world. And indeed, talking about how it spread, there is some indication of who the first person was. Apparently, a small child who was playing with a dead monkey, and then how it was spread to different parts of the Western Africa. So the tracking has been possible because of genome sequencing and faster diagnostics. Of course, the other example of Zika. So just to show you, this causes a condition called microcephaly or small-headed condition. And Zika was originally there in the 1947 and early part of the 20th century in Africa. From there, it spread to other parts of the world. And the latest outbreak that happened was originally in Yap Island. From there, it spread eastward to Brazil during the Olympics. And from there, we also saw it coming to India. So the question is, I talked about these pathogens coming from animals, pathogens coming from wildlife, and you might just be wondering, where do they actually come from? Which pathogens come from which animals? Okay, I've just put a collage of various things that we do with animals. So basically, we grow cows, or this is apparently a prized cow or a bull, and then we have sheep. This is typically the zoo where children can go and play. Of course, a pig pen, poultry, and then we have pets such as cats and dogs. Some people also have turtles as pets, and snakes, and of course birds. And we can get pathogens from many of these. Examples, again, some of the examples that I've given, this is about rabies. So basically, rabies can be transmitted by different kinds of animals in different parts of the world. So in India, it's mostly dogs, but if you look at the Western world, it's mostly raccoon and fox. and also to some extent skunk. So different regions of the world have different animals transmitting rabies. One of the major reservoirs where the animal or the mammal itself does not cause disease, does not see any disease, is the bat. So in bats, you don't actually see the disease, but bats can spread rabies. Other examples I have already talked about. Influenza. So influenza is actually a virus that circulates among water birds. In water birds, it does not cause any disease. And there are various types of influenza viruses, the H types, the N types. I won't go into the details. They can also get these things from bats. But when they jump into species, they can cause disease in various kinds of animals, including pigs, humans, birds. And in some cases, such as the bird flu, they can also kill the chicken. So usually it's not fatal to chicken, but in certain strains of influenza, they can kill chicken. And of course, they can jump to humans, mainly through the transfer to pigs and then to humans. So you can see the double arrow is only between pigs and humans. So pigs are supposed to be what we call as the boiling pots of influenza virus transmission to humans. And as I said, birds are silent carriers, and they may carry different flavors of this. If you wish, you can think about this like different flavors or different colors of ice cream, and you can mix and match different colors of ice cream and produce a new virus. So, I mean, I didn't mean literally that you will produce virus also out of ice cream, but these colors or flavors can mix together to produce a different virus. So that can now be transmitted to humans through pigs if there is a susceptibility or receptor available. And these, as I said, these are typically carried by birds, and from birds they can go to chicken, and from chicken they can come to humans as well. This has been shown with the H5N1 avian influenza. And of course, since the birds are carriers, when they fly to different parts of the world for overwintering and things like that, they can carry the virus without any disease and transmit locally. The H1N1 pandemic influenza, in fact, was a mosaic between a pig virus, which was there circulating before, and another pig virus, a human virus, and a bird virus. So all these together produced a combination of to produce a new virus, which transmitted to humans, for which we did not have any immunity at all, no protection to this particular virus. So, but that's why we had this 2009 pandemic influenza. And of course, I've just shown this. This is not the reason how it spread. Other examples, for example, this was West Nile in the late 90s in New York. Crows were dropping dead from the sky, and people didn't know why. So the reason was the West Nile virus, where it circulates among birds or other animals, and then mosquitoes can spread to humans. So in humans, it causes brain disease or encephalitis, and also in horses. Nipah virus is another example. You may have heard about this a few years ago. Basically, there were some examples in Malaysia where there was disease that was basically tracked back to bats that were on certain trees, and bat droppings were being licked by pigs, and the virus amplified in the pigs. And for those, especially butchers and other people who were in very close contact with the pigs, picked up the disease. So there was mild disease in pigs, and 93% of the cases in this instance was meat handling. The bat excreta were picked up by the pigs. And this is basically in Kerala, I believe 2018, where there were 10 or 12 cases of Nipah, although we do not know what the source was. There's another example of monkeypox. This happened in 2003 in the United States, where there were 71 cases, 35 were confirmed, and 18 were hospitalized. And the reason for this was somebody in Texas imported these giant Gambian rats from Africa, and they were kept together with these animals called prairie dogs in pet shops From there, somebody picked up these pets to go to this state called Illinois and Wisconsin, where people started having diseases, and it took them some time to figure out what this was, because they started observing these wild prairie dogs. These were kept as pets, and they saw that these prairie dogs were dying. So then they put one and one together and identified that this was due to monkeypox. Of course, the hot topic of today, these days, is the origin of human coronaviruses. So human coronaviruses, there are quite a few human coronaviruses, including these that are never talked about. But the main ones that we know now is COVID, or SARS-CoV-2. Although its close cousins such as SARS-CoV-1 and MERS-CoV have been there before, about 10 or 15 years ago. Almost all these coronaviruses came from animals, and potentially their origin was from bats or from mice and rats. So there was an intermediate host, and in the case of SARS coronavirus, one potentially talked about intermediate host is the pangolin, but we do not know if this is exactly the case. We don't have complete concrete evidence. But in a nutshell, we can get all sorts of diseases from animals, from various kinds of animals, including migratory birds, crows, snakes, turtles, and antelope, fox for rabies, of course, pigs. And this is just, this is mostly talking about bacteria and viruses. There is a huge list of parasites where animals are involved in transmitting these to humans, either through certain intermediate hosts, such as snails or other soft-bodied animals, or directly. And bats are a major source of mostly viruses coming into animals, and somebody coming into humans. And somebody made a heat map of how many bat viruses are shared with humans. And you can see in tropical countries, this red heat map shows the number of viruses that are shared with humans. There's a huge number of viruses that are transmitted from bats, and we do not completely understand what keeps them in check in bats but causes disease in animals and humans. So the question is then, how do we actually address zoonosis? How do we address the transmission of disease from animals to humans? This is a very complex topic that is being clubbed under what we now call as One Health. And how did this One Health start? So in the early 17th century, Pope Clement XI actually called a medical doctor, Dr. Giovanni Lancisi. And at that time, what was happening was, this is of course a picture from 1896, not from the 17th century. But the animals, if they have this disease called rinderpest, they die in swarms like this. They have severe pneumonia and diarrhea, and they basically die suddenly. So he was asked to do something about it, because the animals were being used as human food supply. Food supply was getting short. So the popes asked Giovanni Lancisi, do something about this. So after some time and after tracking all this, what he said is, destroy all ill or suspect animals. So basically, if something comes ill, just destroy it so that the disease does not get transmitted further. So this was a milestone in contagious disease control and kicked off an important aspect of control of infectious diseases. And of course, another great contributor was Rudolf Workhoff, who lived in the 18th century. His father was actually a butcher, and he himself started the earliest meat inspection program and studied the life cycle of a tiny parasite called Trichinella spiralis in pig muscle. This is actually a parasite that can come to humans from pig meat. And he also studied cysticercosis and tuberculosis in cattle. And the main thing is, he said, between animal and human medicine, there are no dividing lines, and nor should there be. This was, remember, in the 18th or the 19th century. So, and he was the one who coined the word zoonosis. So what is One Health? One Health is something that considers the relationship among humans, animals, and the environment. And it considers pathogen transmission as being influenced by changes in the environment that humans and animals share. So important thing is, environment, humans, and animals are considered as a single thing in order to understand the diseases. And it's a holistic approach, but it's much easier said than done. So what causes the spillover of diseases from animals to humans? There are a host of factors, too many to list, in fact. This could be socioeconomic factors, agricultural practices, intrinsic factors such as age, pregnancy, even nutritional status, extrinsic factors such as drought, habitat loss, bushfire, cave destruction, urbanization, cultural practices. For example, there is this practice of touching the dead body in Africa, which was responsible for spread of Ebola. Ecotourism, people touring, the forests, international travel, trade, all these things, and domestication of animals, all these things can contribute to spread of diseases to humans. And the greatest burden of these infectious diseases or zoonotic diseases falls on 1 billion poor livestock keepers in the world. And if you see the dots here, they are mostly in Africa and parts of Asia. And the diseases can cause 2.3 billion illnesses and 1.7 million deaths every year. But we have to understand that most of these reasons or most of the drivers of these reasons are anthropogenic, meaning they start with us. We humans are the reason. We have a huge population explosion because of which we are occupying spaces. There is a high degree of coexistence with animals. In fact, 70% of the rural poor are linked to livestock production. There is huge amount of poverty and malnutrition, which leads to problems with food security, leading to intensive farming practices for which we encroach forests and the habitat of wildlife. And there is also ecosystem degradation. And on the other hand, there is lack of infrastructure in terms of inadequate water and sanitation. And because we do not, especially in India, we don't process the food. We have basically used raw. We don't have storage facilities, because of which, we have to cook the food as soon as possible. Weak public health and difficulty accessing care in many places, especially in Africa and Asia. And of course, international travel and tourism. And added to this is the natural phenomenon coming from the environment, such as flood, wildfire, and drought. So the way forward is really to understand and consider together all factors leading to disease emergence, improve data collection, analysis, and sharing. We need to really empower communities, make them understand what these are, what are the factors, how to monitor, and how to respond. Because we cannot do this sitting in the cities. We have to work together in various sectors, and that includes you all, your family, your friends, and the people in your community. So please mask up, wash your hands, keep your distance. We are all responsible for controlling the disease, or if we don't do this, spreading the disease. So it's important to note that we are not in isolation in this environment. We share this environment with pathogens and animals, and there could be a crisscross between all of these three, and we can have diseases or pathogens jump from one to the other. But the problem in implementing One Health is that it is multisectoral. You can imagine any of the things that I've listed here could be brought into one aspect of One Health or the other, including law and legislation. And if you see the lines here running everywhere, each thing influences the other. So basically, it has to be a very highly coordinated effort, and we have to begin somewhere. So we need to move from the traditional approach where we study humans, animals, and environment separately into a One Health approach where we study them together. We have to move from disease treatment, looking at illnesses as symptoms to just to cure the symptoms, or discipline-based research to treat specific illnesses, or just attention to parts or symptoms, or a reactionist science, to an approach where we create health as a whole, health as an adaptive change to be resolved, and a transdisciplinary or an intercollaborative system of research and attention to the wholes to go to the root cause and the system with a proactive science. This is easier said than done. So with that, I will stop my talk and take any questions if you have. So I thank you so much. Thank everyone for providing this opportunity to put across my thoughts, and I'm open for questions.
Now the topic is open for discussion. If you have any questions, please ask. First, I must say it's a very wonderful talk, sir. I think student must have at least identified few terminologies like maladies, zoonosis, One Health program. So don't get confused maladies with melodies, first of all. So, and he has covered almost all important viral diseases which are important from the genetic point of view. And he also talked about genome sequences, how it helps to track back the origin of disease or infection. This is very, very important in the current context from COVID point of view. As you mentioned, that genome sequences, how it helps to identify the different variants of coronaviruses. For example, South African virus, UK variants, Brazilian virus, and now a double mutant Indian virus. And how this One Health will help to overcome these problems.
Sir?
Yes, please.
Sir, good morning, sir.
Yes, please go ahead.
This is Kirtika from CMS College of Science and Commerce, Coimbatore, sir.
Hi, Kirtika.
Sir, I have a question that does COVID affect birds? And is there any possibility for the transmission of COVID between birds and humans?
No, we have no evidence that the virus is transmitted through birds. So far, the only domesticated animals which the virus has been shown to infect are cats and a particular kind of animals called minks. And of course, ferrets. Minks and ferrets are closely related animals of a Mustelidae family. They are kept as pets and also they are used for research. Minks, basically because people use them for making fur coats. There's a huge industry in Denmark. So very recently, in several farms in Denmark, the minks were infected and they basically killed several thousand of minks to stop the spread. There was also evidence that some minks transmitted the disease or transmitted the virus to humans. And as far as cats are concerned, there have been examples of cats that were in close proximity or staying in the same house as some of the people that got infected. But there is no evidence of virus transmission from cats to humans. So as of now, what we know as far as the household scenario is concerned, there is no pet that can transmit the virus to humans. And there is no evidence that the virus can even actually replicate in birds.
Okay, sir. Thank you, sir. Sir, I have one more question.
Yes, please.
Can we use genetic mapping to identify from where contagious disease started?
Genetic mapping of the organism?
Yeah,
you mean
sir.
the pathogen? You mean the pathogen?
Yeah, sir.
Yeah, that's exactly what I was saying. So we can use genetic mapping. In some cases like viruses, we can do the whole genome sequencing because the genome length is not too big. In the case of certain others, in fact, even with certain viruses, we can use actually fragments of a particular genome and try to figure out where they are moving or what is happening. But it is difficult to actually predict whether something causes a huge disease or a pandemic, because most of the diseases or in medicine in general is mostly reactive. For example, if you only take medicine for headache, if you have a headache, You don't take a preventive medicine for headache. I'm just giving a general example. So until there is a disease, we don't try to actually treat it. In fact, there's a famous saying, don't fix it if it ain't broken. So basically, the medicine is mostly reactive, but we can do surveillance in animals, but it comes at a huge cost. Who's going to do it, and who is going to fund it, and what are the diagnostic tests? What samples do we take? From where do we take? So that becomes a big issue. And it's, as I said, it's easier said than done.
Thank you, sir. Sir, this is Kirtika again.
Yes, please.
Sir, can we use 2DG drug for cancer treatment?
You mean that DRDO drug you're talking about?
Yeah, yes, sir.
I am not a clinician, so I cannot really comment on it. What I have read is what you have read in the newspaper.
Yeah, okay, sir. Thank you.
Hello, sir. Good morning. Sandeep here.
Yes, please.
Sir, I have a very, very general question about this COVID SARS-CoV-2 virus. So, of recently, they found that it's been spreading in these Asiatic lions in zoos and all, right? So, my question was, is our domestic dog safe? I mean, are they also been affected, or they show some kind of symptoms? Since you said that it moves from human to animal, but not vice versa, are our pets safe at home? Just curious.
As I said, the highest amount of virus load in terms of animals happens to be in cats. And there have been some cases of some symptomatology in cats. I am not aware of any cats dying of this virus.
Okay.
And most of these cases are that the cats have picked up the virus from humans, not the other way around.
Right, sir.
And talking about dogs, there have been quite a few reports of serological positivity in dogs, meaning you see antibodies in them. So they were infected at some point.
Right,
But virus isolation has not been done, I believe, has not been done from dogs.
Okay. Okay. So that's very interesting that the virus is infecting, but they're not causing any pathogenesis or any kind of symptoms.
Yeah.
Yes. Thank you so much, sir.
Sure.
Sir, I have a question.
Yes, please.
Sir, these days people are rampantly using Ivermectin to as a prophylaxis or maybe treatment for SARS-CoV-2 infections. So, what is your take-home message on using Ivermectin for such infections?
Okay, so ivermectin is a general drug that has been used for a long time as an antiparasitic. And its mechanism of action is to basically block transport of certain things at the nuclear cytoplasmic interface. It is, to some extent, it can block the virus replication, but there is a narrow window during which it works. So prophylactic, in the sense, how long do you need to take becomes important. So if you want to use it prophylactically, therapeutically, there is a short window during which it is useful. I don't think it's useful beyond a point where you need oxygen or a ventilator. So it has to be at an early phase of infection. And same is the case with remdesivir. The only thing for ivermectin is that it has very few side effects and a very safe drug. So that is the major reason that it is being touted as something that you can take. And in fact, I think the government of Goa is saying that they will give ivermectin to all adults.
Sir, I have a question.
Sir,
Yeah, go ahead. Go ahead.
this is Madhuri here. Can I go ahead, Dr. Day?
Yeah, go ahead, please.
Sir, your talk was fantastic, actually. So I just wanted to ask one question. If there is a zoonotic disease outbreak, what do you think the general steps that need to be taken before it becomes a a pandemic? So what do you think it needs to be done?
Okay. I guess there are, first of all, we need to make a distinction about whether it is going to be fatal or not. And the measures that you undertake will depend on how much of severity it will cause. So if you really think it's a zoonosis and can spread fast, containment is the main issue, and this has been outlined with the bird flu incidences, where you have a 10-kilometer radius inside which you kill all the birds, and then you don't allow anything to be transported outside. So there is a whole slew of measures that you can take depending on the scenario. So basically, you have to start treating if there is already something similar available. All the people that are infected, you will have to possibly have all sorts of PPE and all these things that we are doing with SARS-CoV-2. You may have to restrict movement of people as well as animals. Probably also, if possible, you may have to, in some cases, for example, in case of poultry, you will have to cull the animals and birds if that is a possibility. A whole lot of things need to be thought about, but it all depends on whether this is an already known virus or a relative of an already known virus or a completely new virus. So the steps that you take will differ with each kind of pathogen, because if it's a completely new virus or a completely new pathogen which you have no clue about, obviously your steps will be very stringent, and you will have to wait until you identify and start discovery programs on treatment or therapeutics and things like that.
Yeah, so for all the participating colleges, students, as well as faculty members, please throw your question. We are getting very few questions from the students. Please drop your shyness and put forward your question. Anybody?
Good morning, sir.
Yeah.
So this is Dr. Suja from CMS College of Science and Commerce, Coimbatore, sir. First, let me thank the organizers and the speaker for having such a wonderful webinar, which is the need for the hour. Sir, my question is like the diseases associated with the cats, specifically the domesticated cats. Sir, how we can prevent this? And I would like to know is the hair, cat hair, how it is harmful to the humans?
Good afternoon, madam. Just about over afternoon, I guess. As far as COVID is concerned, I will answer COVID as well as other pathogens. As far as COVID is concerned, we don't have evidence so far that the infected cats transmit the virus to humans. That is the first thing, unless probably they become severely symptomatic. Secondly, there are certain other pathogens, including the one that Dr. Abhijit works on, Toxoplasma gondii, that can be transmitted to humans. How you can stop transmission is a difficult thing, because if you are keeping cats as pets, then obviously you can't do much about it. And sometimes you won't even know whether the pathogen is lurking in the cat. The main things are sanitation, 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 sanitation. We cannot stress enough on sanitation. So if you touch the cat or if you hug the cat, just go, clean yourself or take a shower or whatever it is. Cleanliness is, and also you need to keep the cat clean. If there's any vaccination that needs to be done, you need to get them vaccinated. So these are most important things. And as far as hair is concerned, I think you may be talking about allergies. So allergies either due to certain things in the hair, certain components in the hair, whether it is proteins or certain things, or presence of certain tiny parasites such as mites that can cause allergies are a problem, not just with cats, but a lot of pets. So main thing again is cleanliness, both of the owner as well as the pet. If there is, especially for mites and things like that, if there are medications, you can use them. And if you have children, take care that they don't come in contact with some of these medications. Take them to a veterinarian, get them treated. Main thing is cleanliness, sanitation, both of the animal and yourself.
Thank you, sir.
Good afternoon, sir. So this is Anjali Bhattacharya from CMS College of Science and Commerce, Biotechnology Department.
Yes, please.
Sir, my question is, most vaccines that are out are against like the single-stranded SARS-CoV version. But how effective would it be in a community which is mostly affected by the triple-stranded one?
I didn't get the question. How, if triple what?
Triple-stranded variant, sir. So how would the vaccines be more effective?
Oh, you are talking about the variants. Okay. So the evidence is just coming out that almost all of the vaccines that are being used are at least partially effective against most of the variants. The currently used vaccines. It is quite possible, I'm not ruling it out, that there may be some variants that may be completely unaffected by the current vaccines, in which case we may have to come up with we may have to use the new, genetic information from the new variant and include it in the vaccine. That is a possibility, and that is a problem that we have to face with this virus as well as other viruses, including influenza. So, but as of now, most of the vaccines appear to provide at least partial protection against all of these variants, whether it is Brazilian, UK, South African, or the double mutant variant. I don't know if the triple mutant, which one you're talking about and how pervasive it is. So it depends on whether it is spreading too fast, or is it going to die out ultimately to be taken over by, let's say, the double mutant. But I think most of the vaccines are fairly effective.
Okay, sir. Thank you, sir.
Sir, since you are discussing about a vaccine, so I have a question out of curiosity.
Yeah.
So there are some side effects which are associated with Covishield, that the blood clots. So there is a lot of skepticism whether we should take the Covishield vaccine or not. I know it does give a partial protection, but whether if you get vaccinated with Covishield, should we go for any heart echo, because these clots are dangerous whenever they are there in the heart or in the brain. So should we go for some normal echo, ECG or heart echo? What do you suggest?
Okay, so yes, there has been quite a bit of news about blood clots associated with Covishield. But as far as I know, scientific evidence suggests that the frequency of blood clots is not much higher than the normal population. The problem with the early studies was that they were only studied with the vaccinated individuals. Nobody went back and looked at what happened to the general population. So as a little bit of data came out, it's been shown, which was originally done in Europe, that the frequency of blood clots is not significantly higher than the normal population. That is the first thing. Second thing is, it is completely impractical to go and get an echo or whatever other test you want to get done. Basically, because at this time point, the healthcare is completely overwhelmed dealing with people who are sick. So you are going to basically plug that pipeline of healthcare throughout the country, and that is not going to be useful. When there is no strong scientific evidence of blood clots creating problems, I don't think we should be worried about it.
And does it relate to all adenovirus backbone?
That I do not know. As of now, this has been shown only with the chimpanzee adenovirus, which is that Covishield is made of. The Sputnik 5, I don't think there is any evidence of blood clot. I have not seen any reports. So So I don't know whether I can say that it is associated with the chimpanzee. I don't know, but it doesn't appear to be so. Or then again, I'm not certain.
Thanks.
Hello, sir. This is Sunny from NIB.
Yes, please.
Yes, sir. I have a question that means I want to ask you. What do you think? How long this COVID will last? Means when this pandemic is going to be over, and how it differ from the Spanish flu in 1918? Because whatever I read, it lasts for 2 years, 1918 to 1920, and without any vaccination. Because that time we don't know that social distancing and this mass vaccination, and the disease vanished in 2 years. So how it differs from this COVID-19 from the Spanish flu, and how much? What do you think? How much time it takes to vanish the disease?
Okay, so we are already into 1.5 years of COVID.
Yes.
So I would presume, if you ask me personally, I would presume it will take another 1.5 years by the time we have it completely under control, but it is not going to vanish. Influenza has not vanished. It just keeps coming. It's just that we have forgotten about influenza because it does not cause serious death. And it keeps coming, and people are vaccinated. Elderly people, especially in the Western countries, are vaccinated. So they have a certain level of protection. Only thing was in those 1918 pandemic influenza, there were, if you see some of the old reports, there were 3 or 4 waves. And I believe there also the second wave was the hardest. And I think there were fewer people around the world at that time. The virus simply ran out of people to infect. But now, with a huge number of people around the globe, and the staggered distribution, a spread of the virus through different parts of the globe, it may take a little bit more time. So I would think maybe the end of 2022, maybe 2023.
Okay, sir.
So, Abhijit, I think now we are nearing to the completion of the time schedule of the talk. So we can conclude here. Abhijit, go ahead.
Oh, okay.
Yeah.
So if there are no more questions, I really thank Dr. Hegde for his wonderful talk. The question and answer session was really wonderful. It was really informative, sir. And if you want to, for students, I'm saying, if you are really interested in his research, please look into his research areas. You can find that details on our NIB websites. So, and as I mentioned earlier, we also have this 6-month dissertation program. If you are interested in his research, you can apply for the 6-month dissertation program under his guidance. Also, if you are interested to do a PhD in his lab, we have the PhD program, provided you should have a fellowship for 5 years or so. So if you are interested in his research, please apply for a PhD program under his guidance also. So thank you very much, sir. Thank you.
Not just my lab, anybody else in NIH.
Yeah.
Thank you very much.
Thank you. Stay safe, stay healthy. Thank you.
Yes.
Okay.